All right, let's see how it works here. Neuroscience. And we're going to assume you are a non-neuroscientist. I'm not a neuroscientist. I'm a physical therapist, clinician. So we're going to go with that focus as we're clinicians. We need to know enough about this to be able to talk to the doctor, talk to our patients, and do good treatment. So you guys are going to tell me a little bit about yourselves, just a little bit about me, so you know where I'm coming from. This is my old house back in San Diego, California, back Poway. It's a little town about 15 miles from the coast. Kind of a neat quote by Santiago Ramon y Cajal, who's a very famous neuroscientist. He actually was really instrumental in looking at even some of differentiating some of the cells, especially the cells, Purkinje cells, and also the cells in the cerebellum. So anyway, as long as the brain is a mystery, the universe, the reflection of the structure of the brain, will also be a mystery. And it really is. Our brain still is very much a mystery. We're getting better, but the more we find out about it, the more we realize we don't know about the brain. Neuroscience, relatively new science, concerned with lots of different things. It's a combination of the development of the nervous system, chemistry, structure, function, pathology of the nervous system. You know, there's different areas of neuroscience: molecular, cellular, systems, behavioral, cognitive neuroscience. Some of the stuff is just so fascinating, especially the cognitive behavioral stuff. I really wish I had more time to kind of get into some of that and listen to some of that. There's actually a podcast I just heard about that's really kind of focused on the mind, and kind of hoping maybe I can listen to some of that while I'm on the treadmill and that kind of stuff. So, but we're going to be focusing on really the clinical side of things, and really more of the clinical sensory motor neuroscience. So that's really, we're heavily focused on the motor systems, sensory systems, how they work together, generate movement, adapt to, and what happens to them when they're injured. Okay. So organization and plan for this course. You'll notice when you look at your schedule, it's kind of broken up into these blocks of different areas, and that's kind of how your textbook breaks it up into this. We'll start with the cellular level. We're going to do a little introduction, a big overview today, and then we'll kind of move into just the neuronal, cellular type of level functions in the next session, couple sessions. And then we'll also go into development of the nervous system, which will overlap a little bit with maybe some of your maturation science stuff. Is this maturation science? You talking about development of the fetus and those kind of things? And we'll get a little more into just the nervous system development, a little more detail than you're probably getting in that course, and how it relates to adult nervous system specifically. Then we'll get into more of the functional systems. Then we'll break it down by region of the nervous system: brainstem, spinal cord, cerebrum, those kind of things. Then special functions like vision, vestibular, okay, are the two main ones we'll look at. And then support systems, blood supply, coverings, the meninges that cover and cerebral spinal fluid, those kind of things. So they kind of build on each other. Neuroscience, like anatomy, is really like learning a language, just getting the terminology and really understanding that. And as it builds, it gets easier because you start to understand, and we can talk the same language as you start getting introduced to all these different terms. Okay. Let's see, few fun facts, some neuroscience. I updated a few of these just last night because I was bored with the other ones I had on there. Human brain, about 3 pounds. Wasn't there a show? I think there was a show like one time called 3 Pounds or something like that. It was about brain stuff. It was a medical show, but it didn't last very long, obviously. Makes up about 2% of our body weight, total body weight, but about 30% of our blood supply is going to support brain. You know, our brain just functions on, oxygen and glucose, and it doesn't store any of that stuff. It's like it needs it all the time. If you lose blood supply or oxygen or glucose to your brain, you pretty much are out pretty quick, okay? So that's important there to realize, and that's another reason why, for me, with my less hair nowadays, putting a hat on, I'll sit there and be freezing, like in my house, or I'm getting cold all the time, I'm just getting like an old man, and I go, just put a hat on, like no wonder, because it's 30% of your blood supply is going up there. So the brain itself doesn't feel any pain. So once you get past the coverings, like the dura mater, this tough, thick cover, that has pain receptors. So that's when people have really excruciating, sometimes headaches. It could be because of pressure on the dura or tension on the dura. But the brain itself, once you cut the dura open, get through the skull and the dura, you can poke the brain, move it around, there's no pain. And you've seen somebody on shows like the surgeries where they'll like go in and maybe somebody has seizures or something like that, and they'll open up, they'll put them out, put them in a light anesthesia, they'll do the craniotomy, cut through the dura. Then once they do that, they wake them up again. And they'll go in there, like if they're trying to do like a stereotactic surgery, or they're trying to like lesion a certain area of the brain, like where maybe the epilepsy is starting, but they don't want to affect the person's function, like especially like verbal skills or motor skills and those kind of things. They'll be moving this little needle around that they can cauterize or lesion that site to try to reduce the seizures, but they don't want to hit a spot that might cause functional deficits. So they'll have the person talking, or even they'll have them like bring in if they're like a pianist or a violinist, they'll have them actually play their music while they're messing around with their brain. And like if they notice a problem, like, oop, we don't want to hit that spot. So pretty cool. We have about 100 billion neurons, okay? And if you actually laid those side by side, not even end to end, like with the dendrites and the axons all end to end, just side by side, you go about 600 miles laid side by side. That's what's compacted into our brain if you put those all laying side by side, which creates over 1 quadrillion synapses. So that just talks, when you think about computing power, our brains are just amazing, the computing capacity they have. 500 million synapses or contacts per cubic millimeter. Cubic millimeter, it's insane. Most of the cells, though, in our brain are not neurons. So you have 100 billion neurons, and that only makes up about 25% of all the other cells in your brain. So all the other cells are support cells, like glial cells we'll talk about, okay? Brain freeze, you guys have all had brain freezes, I'm assuming, most of you at some point in your life. If you haven't experienced it, good for you. I just thought it's called sphenopalatine ganglioneuralgia. So next time you're out and somebody has that one, you can pull that word out and say, how's that sphenopalatine ganglioneuralgia doing? So it's actually what causes vasoconstriction. Okay, your body senses that there's it's sensing a rapid drop in your core temperature, even though you're not really having one. It's just because it's hitting a sensitive spot in the back of your palate there, and so it causes a rapid vasoconstriction, and it causes a local rapid vasoconstriction, even though it really tends to be a more mass one, because when you start to have a drop in core temperature, you vasoconstrict your extremities so that your blood gets shunted to your core to warm it up. But it's kind of your brain gets confused briefly, and it causes vasoconstriction regionally initially, just right there in your head, and causes that feeling of a headache. That pain is because of rapid vasoconstriction in the vessels right around that area, in that kind of sensitive area there. So you guys heard the term, we only use 10% of our brain, that kind of stuff. That's really a myth. We use most of our brain most of the time. I wish it was true, because then I would have hope that I could be smarter. But we use different regions more so than another, but our brain is always working, and most parts of the brain are working somewhat. It may shift, but there may be a time when it's only working 10%, but most of the time we're using more than 10% of our brains. So there is some interesting data, though, to show that if you're standing and you're moving, your IQ is actually elevated by about 10 to 20 points if you're more active or standing and those kind of things. So that's why I stand and move around. I need all the help I can get, so I stand up, move around a lot. So hopefully IQ, come on, make me smarter. But if you think about where the prefrontal cortex, which is kind of your executive functioning, personality, what makes you and making decisions and those kind of things, the thing that's closest to it is the motor strip, okay? Which is your primary motor strip and your movement. And so movement is very intimately tied to the prefrontal cortex. And what they assume from an evolutionary perspective is that when an organism learned to move and explore the environment, that's what developed the prefrontal cortex, the problem solving, and how do I interact with my environment? I can move in my environment, but now I got to manipulate my environment and interact, and that's only through cognition and those kind of things. So So it's interesting kind of when you talk about exercise and how that improves cognition and thinking ability. That's one of the reasons why they think it does, because they're intimately tied to each other, motor system and cognitive functioning. All right, let's get through a couple things here before we take a break. Starting basic. What are the major components and organization of the nervous system? I think I have this attached to your back of your handout there, the last page of that handout. I like to see the big picture of things, and it helps me kind of understand all the different little parts and how they're related to each other. So this is the back page of your outline that I handed out, okay? So you can turn to that. So this is this big picture today that gives you some of the key areas or components of the nervous system, and it's kind of how we're going to talk about it. So we have the nervous system on the left there as a whole. We can break it down into some different subcategories. So we have basically the neurons and the neuroglial cells, so the cellular level. We can kind of artificially break out this thing called the autonomic nervous system. That's how your book does it. The autonomic nervous system has elements or parts that are both in the peripheral nervous system and the central nervous system. We're just going to call it out as the autonomic nervous system, but just understanding that it does have elements that are in the peripheral and central nervous systems. But then we have the peripheral nervous system. We have the central nervous system. Okay, and if you start following that little chart or graph or line diagram out to the right there. You know, if you look at the autonomic nervous system, we can divide it into a sympathetic and parasympathetic component. If we look at the peripheral nervous system, it has cranial nerves are part of the peripheral nervous system and spinal nerves, okay? We could keep branching that out, just like I've seen some of you guys do these crazy drawings on the boards of the blood supply or the nervous system, but to the brachial plexus, to the individual nerves. So you could just keep taking that out. But right now, today, let's just start with this, okay? So peripheral nervous system is mainly your cranial nerves and your spinal nerves that are going to go out from your central nervous system. Then here's your central nervous system. We can start by starting with the brain and spinal cord. Then we can take the brain and we can divide it into cerebrum. We have a cerebellum and we have a brainstem. Then we can start subdividing the cerebrum into different categories and keep just following that out. We're going to talk about all these elements, but that just gives you an idea in one picture, like how is all these things related to each other? What's a subcomponent of what? And these are just the major subcomponents of different structures and different regions of the nervous system, okay? So starting really basic here, okay, which is these ones right here, neurons, ANS, PNS, CNS. So that's what we're talking about here. We have the neurons and neuroglial cells, the central nervous system, peripheral nervous system, and autonomic nervous system. Neurons, okay? These are the things that make it happen. Basic structural functional unit of the nervous system. These are the things that actually, you know, have properties like the ability to store a charge and the ability to change and alternate and create an action potential and send a signal and propagate a signal in a directional way, okay? So those are neurons. They convey information if a neuron is bringing information into the central nervous system or towards a structure within the nervous system, it's called an afferent neuron. So afferent. And I think of the A, I always think acquire. It's acquiring information. Afferent is acquire. It's bringing in information to that region, to that structure, to that part of the central nervous system is an afferent neuron. Okay, so when we talk about sensory neurons, like sensory neurons that come like from my hand and go up to my spinal cord, those are afferent neurons, bringing information towards the spinal cord. Okay, or we may have a certain part of a brain that's information is going from one region of the brain to the thalamus, and that's an afferent neuron. It's taking information towards another structure. Okay. Then we have efferent neurons. Efferent neurons are neurons that convey information away from the central nervous system or away from another structure within the nervous system, okay, going towards another structure or another system. So motor system, okay, so we have nerves that go, just like we have nerves that take information towards our central nervous system for sensory, we have nerves that take it away, go from the central nervous system towards muscles for motor, okay, and those are efferents. And I always think efferent is exiting. It's efferent. The E is exit or goes away from that, you know, taking information away, a signal away from it. Okay. And then we have neuroglial cells. Remember, neuroglial cells are actually more numerous than neurons. Okay, these are all the support cells that make up a big part of the mass of your nervous system, especially your central nervous system. Okay, so glial cells are non-neuronal cells. They do not convey information in the same way a neuron does. They don't propagate action potentials. Okay, they may have some fluctuation in charge. And they may have certain molecules that move and certain ions that move between them, but they don't generate an action potential per se, okay? But they actually do contribute now, as more and more research comes out, these glial cells may contribute to information processing more than we thought they did. They're not just totally dumb, innocuous non-members of, they're not just purely support cells. They actually can sometimes help, enhance a signal, cause some inhibitory effect in a region, but they don't generate action potentials. They're primarily there to insulate, provide insulation. So things like Schwann cells that wrap around the neurons, your axons in your peripheral nervous system, provide insulation, just like on an electrical wire, that little plastic, that's an insulation. In the central nervous system, you have these things called oligodendrocytes, which are a type of cell that do the same thing as a Schwann cell in the peripheral nervous system. They wrap around the axons of the neurons, so you send that signal more effectively. You don't lose that signal, that action potential, by insulating it. They provide some nourishment, okay? So they do have stores of some like glucose, a little bit, not much, but the little very brief stores. They help to support. They actually provide some support. They form the meninges, the dura, arachnoid, and pia layers that surround your nervous system, so they provide some protection of your nervous system. The glial cells are where most central nervous system tumors come from, because glial cells are much better at reproducing themselves. Neurons used to think neurons didn't reproduce. Once you were in adulthood, your neurons, you only had as much as you had, and then if they died off, you never got new ones. That's why, I don't know if your parents or teachers, don't drink, you're killing all those neurons, you'll never get them back, kind of thing. But neurons are actually a little more resilient, and there's actually some reproduction of neurons even as we get into older age. Not nearly, though. Neuroglial cells, they're always reproducing. Like a lot of our tissues, our skin cells, our liver cells, those kind of things, where they're turning over, okay, and they're reproducing. But that's why cancer is typically just a defect or a problem with this turnover or reproduction. It's over-reproduction. They goes haywire where you just produce too many cells that you don't need them, and they're non-functional. Plus, if they're in your cranium, they take up too much space, and you get pressure built up in there too. Okay, so when you hear like a glioblastoma, glial cells, it's because of over reproduction of the glial cells, and it causes this tumor. Okay. Let's do the 7 main parts, and we'll take a break. So we're just talking about central nervous system. Let's go through central nervous system. We started with neurons and neuroglial cells. Now we're talking central nervous system. Your book talks about 7 main parts of the central nervous system, so we'll go with that. It talks about the cerebral hemispheres, okay, as being one main component. The cerebral hemispheres includes some big structures deep inside of them called the basal ganglia structures, okay. We'll get to those later, just to know that those are deep embedded within the cerebral hemispheres. Then we have what's called the diencephalon. Okay, the diencephalon, primarily, di is 2, right? So we're primarily talking about 2 structures together that make up this diencephalon called the thalamus and hypothalamus. And those are even deeper than the basal ganglia. They're right in the middle between the 2 cerebral hemispheres, but they're considered separate from the cerebral hemispheres. They're their own kind of structure, this thalamus and hypothalamus, and you have one on each side. These are all paired structures. Okay. Then you have now we're in the brainstem. You have a midbrain, okay, which is the very top part of the brainstem. Then you have a pons, which is the next part of the brainstem. Then you have the cerebellum sits off the back of the pons. Okay, that's another big component of the central nervous system. The cerebellum's coordination, okay, primarily. Then the medulla, which is another part of the brainstem, which sits below the pons, and then finally the spinal cord. Once you exit out the foramen magnum, the hole in the bottom of your skull, that's the delineating spot where you turn from the medulla to the spinal cord. Okay. And so here's those main structures here in this particular picture. And this is looking at a sagittal section through a person like this. And we're looking at the inner side of their right hemisphere. Okay, so we've cut right down. There's this big fissure right between your cerebral hemispheres, the cerebral longitudinal fissure. We've cut right down between it, and we're looking at the medial surface of this person's nervous system, basically, straight down the middle of their nervous system, between the cerebral hemispheres, right down the middle of the brainstem, the middle of the spinal cord. So we're looking at what's called a mid-sagittal section through all of those structures right here. And then we're just looking at a very kind of simple line drawing of that. And so just defining those, let me see if this pen gets kind of ornery on me sometimes. See, that pen is not supposed to do that. And go back to, I just wanted to change colors. All right, here we go. So this structure here, this is all the cerebral hemisphere, okay? Deep inside there, which you can't see, sitting deeper in there is the basal ganglia structures. So those are part of that. Then we have the diencephalon, which is this pink area right here, which is the thalamus, which is really this kind of egg-like area here, which I'm kind of artificially defining the borders of for you. And then this is down here, lower is the hypothalamus. Okay, hypo, below, it's just below the thalamus. Okay. Now we're in the brainstem, and this is called the midbrain, this part of the brainstem right here. And if you look where it's located, it really kind of almost is in the middle of the brain there. It's really kind of up inside of there, that brainstem. Then this is the pons right here, the next part of the brainstem. Then sitting off the back here, see, every time I write it wants to change slides on me, and I don't know why it's doing that. That's the cerebellum sitting off the back of the brainstem for more coordination function. We'll get lots of information on the it's an important structure for us as physical therapists. And then down here, below that, below the pons, is the medulla. And then here's that foramen magnum. Here's the opening in the bottom of your skull here. And then that defines now where the spinal cord starts. Okay? So those are those 7 main components of the central nervous system. And we'll start talking about peripheral nervous system and all that when you get back from your 7 or minute so break. All right? Any questions? So far, so good. We're back going here. All right. So we just talked about the central nervous system and its 7 main parts. Now we're going to briefly just mention the peripheral nervous system. You guys know more about the peripheral nervous system than I do right now. So just that you have that one up on me right now. You guys know your brachial plexuses, your sacral plexuses. I don't know. It's better than I do, I'm sure. So that may fade quickly. We may be on the same par here after a while, but right now you guys got that advantage. But basically the peripheral nervous system is all parts of the nervous system that are not encased in the skull or vertebral column. Okay? These include your cranial nerves. Okay? They arise directly from the brain or the brainstem itself. So don't freak out on cranial nerves. The cranial nerves are like your spinal nerves. They just come out of the brain and brainstem instead of the spinal cord. Okay, so and they have some specialized functions. Okay, some special like vision, vestibular, taste, that kind of stuff. But then you have your 31 pairs of spinal nerves that you then go out to form your brachial plexuses and then go out to form your peripheral nerve distributions and all that kind of stuff. So basically everything in this picture in yellow is your peripheral nervous system, what you learned last semester. Then we talk about the autonomic nervous system, and they kind of artificially pull this out because it really is a feature of both the peripheral and central nervous system. It has parts in both of those areas. But the autonomic nervous system, this is basically our sympathetic and parasympathetic nervous systems that make up this autonomic nervous system. If you remember from physiology class or whatever, the sympathetic is that fight or flight. It generally kind of upregulates things, increases heart rate, respiratory rate, causes pupillary constriction, raises some, those kind of things. So we'll have a whole day on autonomic nervous system. And then the parasympathetic is kind of the opposite effects of the sympathetic nervous system for the most part. Sometimes referred to as the need and feed system. Okay, The sympathetic nervous system, going back a second, is also sometimes referred to as the thoracolumbar division of the autonomic nervous system. So if you hear the term thoracolumbar division of the autonomic nervous system, we're just saying that's a sympathetic division. Where the parasympathetic, if you hear the craniosacral division of the autonomic nervous system, that's the same as parasympathetic. And I'll show you why in a second here, why they use those terms. But what do? They primarily, the autonomic nervous system innervates smooth muscle, okay, as opposed to striated muscle, which is your, move joints and those kind of things. We're talking smooth muscle of the gut or the esophagus and those, or the cardiac muscle, and then secretory glands, okay. And primarily they play a role in homeostasis, maintaining normal homeostasis under different environmental and conditions and exercise and movement conditions and so forth. So it's the automatic system that helps to maintain normal homeostasis no matter what's happening around you or with you, okay? They also play a role, especially in reproduction, sexual function, those kind of things as well. So this is just showing you those two divisions. So here on the left is a sympathetic division, also known as a thoracolumbar division. And the reason they call it is because the nerves that go to supply or go to the different regions of your organs and body that are carrying sympathetic information or sympathetic, control are exiting in your thoracolumbar division or area of your spinal column, okay? So they're coming out with your nerves, and once your spinal nerve forms, your spinal nerve has, I don't know if in anatomy you talk about ventral roots and dorsal roots, so there's roots that are coming out of your spinal cord. Once they join together, all the nerves that are after that are all mixed nerves. All those nerves contain some sensory information, some motor information, and some autonomic nerves, okay? And so they're all mixed nerves. And so the nerves that are coming in that thoracolumbar region have some sympathetic nerve function that comes out of those regions, okay? Then the parasympathetic, also referred to as the craniosacral division, it controls those organs and controls your parasympathetic control through the cranial nerves, okay? And then also a few sacral nerves that are going to your lower gut and your genitals and so forth and bladder, okay? That controls the parasympathetic control is coming mainly from the cranial nerves and from the sacral nerves, okay, that balance the sympathetic nervous system out, but nothing's coming from that thoracolumbar division. You don't have any parasympathetic fibers coming out anywhere in that thoracolumbar region, only in the cranial nerves and the sacral nerves. And the cranial nerve number 10, the vagus nerve, does most of the work for the cranial nerves, is the big one that does a lot of control of heart, lungs, those kind of things, as far as exerts that's parasympathetic control over those. So, all right, so background stuff. Let's start talking language, because this is important to understand. And if you can kind of see how terms are built and some of the root, going back to the roots of these words, you can sometimes decipher a lot about, even if you don't know exactly, can't remember exactly what that term is referring to, if you break the word down and those kind of things, it can be helpful for you to understand that. So we'll talk about that. The first thing we're going to talk about is how to describe locations in the nervous system. You guys have learned, the anatomical positions and the body's anatomical position and the longitudinal axis through the body, referring to kind of this position when things are anterior, posterior, superior, inferior, and so forth. We have to understand that you have the anatomical position and this longitudinal axis of the body, but the nervous system has what's called the neuraxis. And the neuraxis is similar in some parts of your body, but then it changes in other parts of your body. So the neuraxis in your trunk, in your spinal cord, it's in the same alignment as your body's anatomical axis and anatomical reference points. But once you get up into the cerebrum, your brain does this. There's a flexure, there's a little angle in the neuraxis. It tips over like this. So when we get into the brain and cerebellum, it gets a little different as far as what terms we use to call when something's superior, dorsal, rostral, ventral, and which ones are synonymous with which, depending on where we're at in the nervous system. I'll make it way harder than it probably needs to be, but I'll try to, hopefully it'll click at some point for you. But these are the terms. Have you guys learned like rostral, caudal, ventral, dorsal much, or just not seeing a lot? I mean, some of you may have heard it at some point. So here's the terms. You guys have learned superior. It means one part is situated above another part. Inferior, a part is below another part. When we use the term like rostral and caudal, we typically use those more when we're talking about what's referring to the neuraxis versus the body's longitudinal axis, so the nervous system axis, which has that little bend at the top, okay? If you say something is rostral to something, you're saying it's more towards the head or the beak. Rostral is beak, so towards the head or the beak compared to another part of the nervous system. So one nervous system feature or area is more towards the head or the beak of another part. Caudal is towards the tail or towards the coccyx, okay, in relationship to the neuraxis. Anterior, toward the front. You guys already know that, of the body's axis, okay. Ventral, if you talk the word ventral, that's towards the front related to the neuraxis, and we'll show you pictures. It'll make sense. Right now, I'll just talk through the terms, but we'll show you pictures. Posterior, toward the back, as it refers to the long axis of the body. Dorsal is towards the back in relationship to the neuraxis. But there's times you can interchange dorsal and posterior depending on where you're at in the body or in the nervous system. Okay. And then there's a term ipsilateral. That's when one thing begins and ends on the same side. So when we start talking about pathways in the nervous system, if a pathway starts on one side and ends on the same side of the body, like a motor pathway or a sensory pathway, it's called an ipsilateral pathway or ipsilateral tract. There's a few pathways that will cross over, then get down and recross over at the spinal cord. But if it starts on the same side, it ends on the same side, even if it crossed the midline of your nervous system, as long as it starts and stops on the same side, it's an ipsilateral pathway, okay? If it starts on one side and ends on the opposite side, creating this line down the middle of your central nervous system, that's a contralateral pathway, okay? So if it starts on one side and ends on the opposite, contralateral pathway. So let's show you the neuraxis, okay? So in nice four-legged animals like reptiles and crocodiles, their longitudinal axis through their body matches exactly the neuraxis because everything's lined up. So if you look at this crocodile up here, his brain is sitting, why is it doing that? His brain is sitting up here. I have to figure this thing out. His brain, you know where his brain is. And his brainstem and spinal cord, they're all lined up in a line like this that happens to match the axis through his body, right? If you look at a person down here, the axis through the body goes all the way and goes straight up through the head like this. If you're taking just the body's axis right through like this, okay? The neuraxis, though, if you take that axis through the nervous system, as it comes up into the cerebrum, it curves and goes this way because your brain, during development, and we talk about there's a thing called the cervical flexure. Initially, it's all lined up, but as you become matured as a embryo, your brain tips over like this on top of your spinal cord and your brainstem, okay? And so that neuraxis does this, okay, and that has a little angle to it, okay? So if we just purely talk about the neuraxis, which is what these pictures are showing, if you're to the back of that, okay, then you're dorsal. This is ventral here, okay? But as you get up, so this is through the spinal cord and brainstem, it's going up and down, and it happens to match, in this case, the body's longitudinal axis. So dorsal, towards the back, happens to match up with the term posterior here, okay? Does that make sense? So if I'm talking about structures within the spinal cord or within the brainstem, I could say something is, you have things called the dorsal horns, those are dorsal, in the back part of the spinal cord. Or there's these called the posterior columns in the back of the spinal cord. They're posterior columns. Dorsal and posterior are synonymous in this case when you're in the spinal cord and the brainstem. Okay, when you're trying to say one structure, how it's related to another, if it's behind it, it can be dorsal or posterior. We're in the spinal cord and brainstem. Okay, but if we get up into the cerebrum and the cerebellum, okay, those are the structures that are tipped this way. Their neuroaxis is going this way, more horizontal in relationship to the body's axis. So up here, you'll see now dorsal is up here, okay? This is dorsal, so because if you extended that straight up, it would stay dorsal over here, but it tips. So now this is dorsal. So when you're in the cerebrum, you might say, something like the corpus callosum is dorsal to the thalamus. It's a little bit dorsal towards it. And also you could say it's superior. So in this case, dorsal, and in the cerebrum, dorsal and superior, can't write on that, are synonymous. See what I'm saying? When you're up in the cerebrum, where when you're down in the brainstem and spinal cord, dorsal and posterior are synonymous. So all you got to know is when you're trying to read a test question, and I'm saying if one structure is located relative to another structure, just say, if I can use any of those terms, dorsal, posterior, anterior, rostral, caudal. You just have to say are we in the brainstem and spinal cord? If so, then I know that like dorsal and posterior matched up, ventral and anterior are synonymous. Caudal, erase that posterior there, and inferior are synonymous right here. And then we have superior above here, or rostral. Superior or rostral are synonymous when we're talking about brainstem, spinal cord. Those terms can be used interchangeably when you're talking in the brainstem and spinal cord. Let's do that again. So brainstem, spinal cord, because it matches your body's longitudinal axis of the body, okay? So dorsal and posterior equal, ventral, anterior, caudal, inferior, okay, are synonymous. And then superior and rostral. This is going more towards the head or the beak. But now when we get up into and I'm going to go to the next slide here. We go to this slide, it makes it a little bit kind of more easy to see, I think. So here is that cervical flexure, that change in the longitudinal axis through the neuraxis relative to remember the body's axis just keeps going straight up like this, okay? So this is one picture where you can see it all matched up here. So down in the brainstem and the spinal cord here's dorsal and posterior, being matched up here. Does that make sense? Dorsal and posterior. Here's ventral and anterior are the same down in the brainstem spinal cord region, okay? And then superior. This is going to really bother me if this keeps going on. Superior and rostral. This is superior, but remember this curves here, so if you extended that out, that would be rostral. That's just in the brainstem and spinal cord. So superior and rostral. So I could say the C1 segment is rostral or superior to C2. Does that make sense? Okay. Or the midbrain is rostral or superior to the pons. I just don't want the language to get you guys mixed up. But then if we get into the Cerebrum. Yeah. It's gotten worse. I hate when Windows does updates. It always makes things worse, it seems like. So here's cerebrum and cerebellum that are, the neuraxis is running this way, but the body's longitudinal axis is running this way. But so in this case, when we're in the cerebrum, if we're trying to describe structures in the cerebrum, like I say, if I say the corpus callosum up here, and here's the thalamus down here, so the corpus callosum, I could say it's superior to the thalamus, but in this case it's or dorsal. Superior dorsal are matched up here. So the corpus callosum is dorsal to the thalamus or superior to the thalamus because we're in the cerebrum. Or if I go forward, if I say the head of the caudate nucleus is either anterior or rostral to the thalamus. So that's rostral and anterior matched up here. It's interesting. What I'm thinking, what I'm noticing, if I take the pen and go that direction, where if I go to the left, it makes the slide change. But if I go, oh, nope, I thought I had it, thought I figured it out. I thought if I went, oh, nope. Gosh, I'll have to get a new computer. This would be a good excuse to get a new computer, maybe. So rostral and anterior synonymous. Ventral and inferior are synonymous in this region. Caudal and posterior are synonymous when you're in the cerebrum and cerebellum. All right, got it, sort of? If you have questions or need, come to my office. We'll draw it out again. I just, again, I always make that seems like harder than it needs to be. But I just don't want little terminology that they have to trip up a test question. Just where am I at? Most of the time, it's not a problem. Now let's talk about anatomical planes in the nervous system. You've talked about anatomical planes, coronal, sagittal, all that kind of stuff in the body. So it's the same for the nervous system. This is pretty simple. So we have a coronal, which is a frontal, like this. So like take a sheet of glass or guillotine or whatever and slice this way through the front of my brain or spinal cord. That's a frontal section or a coronal section. Sagittal. Sagittal's down the middle, cut down this way, okay? And then horizontal would be this way, okay? So a transverse plane. So here is your sections right there. So it's showing that same thing where your horizontal, coronal, sagittal section is the same as for your body. There's mid-sagittal section, and mid-sagittal is right down the center of your central nervous system, okay? Right down the center of your brain, spinal cord. But then we have parasagittal sections. The only thing I don't even think we show you any parasagittal sections. We do have horizontal sections that we do through different regions. So this horizontal section we might do, there's a mid-horizontal, like these are showing you a mid-sagittal, a mid-horizontal, and a mid-coronal section. We do some horizontal sections that are in different levels of the brain, not just purely a mid-horizontal section. And we do a few coronal sections through the brain that you have to be aware of. Not a whole lot. It's not like we have to know every slice through the brain every few millimeters like neuroscientists or neuroanatomists do, but you need to know a few horizontal plane sections and a few coronal plane sections. We don't do any real parasagittal sectioning. We just usually show you just a mid-sagittal section and in inner surface of the brain, or right down the middle of the brain stem, or spinal cord. So these are those sections. The horizontal section is on that upper left. That's a mid-horizontal section through the brain. Then the next one is a mid-coronal section through the brain, so that frontal view. Then the mid-sagittal section right down the center there. And then this is the horizontal section through the brainstem, through the medulla. This is a section of the medulla down that lower picture below. That's a horizontal section. You'll definitely be seeing some horizontal sections through the brainstem at a few different levels when we get to the brainstem. More terminology, just to kind of keep you clear on this. Terms are pathways for groups of nerve fibers. So these are usually groups of axons, these axons that are parts of neurons, these white matter fibers, right? They're white because they have this oligodendrocyte, that insulation, okay, that forms this insulation that has more of a white-looking look to it, okay? Although it's interesting, when they stain for cross sections, that oligodendrocyte, that white matter, uptakes the dark stain, so it actually is like a black and white reverse image. It actually, when you look at the cross sections through the brainstem, the dark stuff is the white matter. But when you actually look at a cross section through the brain, or you look at the lighter stuff, is the white matter. Okay. So these are some terms you'll hear when we start talking about like things like a capsule. We'll talk about the internal capsule. That's a big group of fibers that runs between the surface of your cortex down through the middle of your brain, down into your brainstem. Is called the internal capsule, and that carries motor and sensory fibers. But it's a capsule of just of nerve fibers, okay, of axons. A radiation, like we'll talk about the optic radiations. Those are the projection of the axons going from a structure called the lateral geniculate nucleus back to your occipital lobe. There's these white matter fibers that are going from the cell bodies located in the lateral geniculate nucleus, and their axons are going back to the occipital lobe, a big group of them, and they're all called the optic radiation. So you hear these term radiation sometimes. A lemniscus. Lemniscus means ribbon-like, and lemniscus is generally kind of a flattened ribbon-looking section of nerve fibers running. When we talk about the sensory system, we'll talk about the medial lemniscus. And you don't have to write some of these terms that I like, medial lemniscus, and I just start telling you, because you'll hear them multiple times. So, but I'm using the term so you understand where you might hear it later on. The lemniscus is what the medial lemniscus carries our proprioceptive information up into our brainstem. Okay, it goes in our spinal cord up the posterior part of our spinal cord, and once it gets into the brainstem, it's called the medial lemniscus because it flattens out. All these axons look like this little flattened ribbon-like structure, and it actually twists as it goes up in your brainstem like a ribbon twisting up into your. So anyway, that's the lemniscus. A tract, you know, we have the optic tract. We have lots of tracts in our. If it's in the CNS, it's called a tract. Okay, that's just another term for white matter fibers, axons. Okay. A fasciculus. Fasciculus it means little bundle or packet. It's a smaller, bundle of axons. Okay, we'll talk about the fasciculus gracilis, the fasciculus cuneatus. These are little pathways of axons that are carrying proprioceptive information from different parts of your body. Okay, then there's a thing called a funiculus, which is actually larger than a fasciculus, and actually a funiculus might carry a couple fasciculuses inside of it. So it's a larger grouping of axons that may be made up of a couple smaller fasciculi, but if you hear the term funiculus, like there's like your spinal cord actually can be divided into like these four major funiculi. There's the outsides of your spinal cord is the white matter, and there's like the anterior funiculus, the posterior funiculus, and the lateral funiculus on each side that carry all the other pathways that are built into there. Okay, so the term funiculus is a larger packet of axons. A nerve. A nerve is basically axons, right? We get out into our spinal nerves, there's a bunch of axons in there. There's motor axons, sensory axons, and autonomic axons, typically in those. Once you get out in the periphery, all your nerves are a combination of axons that are both carrying information towards muscles, bringing information back from sensory system, or carrying some autonomic function. Okay. A commissure. A commissure, like commissural fibers, is where two things join. We'll talk about like some commissures in your brain that connect one part of your brain with another part of your brain, and from one side to the other side typically. Like there's the anterior white commissure. And it's anterior white commissure because it's white matter, it's axons, and it's connecting your temporal lobe on one side to your temporal lobe on the other side, so the two temporal lobes can communicate from left to right through the anterior white commissure. Okay, so if you're commissure, that just means white matter, axons. These are all terms for white matter axons grouped together. Okay, so anytime you see these attached to another term, we're just talking white matter. Does that make sense? A little easier on groups of cell bodies. So when you talk about when you have a bunch of cell bodies that are close together, okay, that are grouped up together, they form these kind of structures. They might be referred to as a ganglia. We have the basal ganglia, and we also have a thing called the dorsal root ganglia, which is out in the peripheral nervous system. And actually, usually the term ganglia is reserved for groups of cell bodies that are in the peripheral nervous system. However, the exception is the basal ganglia, which is part of the central nervous system. but it's still called a ganglia. However, there's a push by some neuroanatomists and scientists to call it is basal nuclei. Because typically if a group of cell bodies within the central nervous system, you call it a nucleus or a nuclei. Okay, and so there's a push to call it the basal nuclei, not the basal ganglia. So just know that. But most people call it the basal ganglia, but you could call it the basal nuclei. If you hear a lamina is a layer, like a laminated board or something like that. It's like these different layers. Well, you can have lamina that are layers of cell bodies. Like when we get into the spinal cord, and the gray matter, the dark matter that makes like it looks like an H in the middle of your spinal cord, that's divided into different lamina or layers. And even in our cerebrum, we have some lamina or layers in our cerebrum that are different types of cells and that form these different layers. And there's the cell bodies, different types of cell bodies. Okay. Then there's a thing called a body, like we'll talk about. Some people call it the geniculate nucleus or the geniculate body. If you hear that term body, a body, especially in the central nervous system, is generally referred. That's a group of nuclei, cell bodies that are together that form this body. Cell bodies form the body. And or the term nucleus. Okay. Nucleus gracilis, nucleus cuneatus. Well, you'll hear these terms. Okay. The nucleus gracilis is associated with the fasciculus gracilis. So the nucleus gracilis is all the cell bodies. The fasciculus gracilis is all the axons coming from those cell bodies. Okay? And we talk about some of these pathways and things. More terminology when we're looking at especially the surface of the nervous system. We're looking at the central nervous system right now, brainstem, cerebellum, spinal cord. If you hear the term sulci, that means rut or furrow. So all the little depressions, ruts, furrows, the little wrinkles on your surface of your cerebellum and cerebrum, those are referred to as sulci. Okay? A fissure is just a deeper, larger rut, depression, whatever. So we have a couple big areas, like we'll call this the cerebral longitudinal fissure. That big area, like I pull my brain apart right there, that big, deep, that's a fissure. Or like right on the side of my brain, there's called the lateral fissure, the Sylvian's fissure, lateral fissure. A fossa, you guys know what a fossa, it's a depression, just like fossas on bones. This is a fossa, that's a depression in the nervous system. We'll talk about the rhomboid fossa in the brainstem, a little depression in the brainstem called the rhomboid fossa. It's kind of rhomboid shaped, Bumps and ridges. So now protrusions or bumps or ridges, we just talked about furrows and depressions. Now we're going to talk about the bumps and ridges. If you hear the term peduncle, we'll talk about the cerebral peduncles. That's a part of your midbrain that connects your brain to your midbrain called the cerebral peduncles. That's a connecting or stem-like connecting part, peduncle. A gyrus, we'll talk about the gyri, and we have the sulci and we have the gyri. The gyri are the actual bumps, on the surface of your brain and your cerebellum. A brachium, if you hear the term brachium, we'll talk about the brachium of the inferior colliculus. Underneath that brachium, if you look underneath, if you cut into that, there's actually white matter fibers running there. It's brachium means arm. It's like a connecting arm between two structures. Okay. A tubercle. Tubercle is just a little bump, a nodule or small eminence on the bump. We'll talk about the cuneate tubercle, the gracile tubercle. These are little bumps on the back of your brainstem. That's all that means. It's just a bump. And usually under those little tubercles, those will be little cell bodies in there. Okay. Then we have layers within the central nervous system. We have things called laminas. We talked about the laminas of the spinal cord. We have a cortex, which is means bark, which is that surface. It's just a couple millimeter or so thick of, on our cerebrum. Most of the cell bodies are just that couple millimeter layer on the surface of our brain. All the deep stuff is mainly white matter, with a few exceptions. We have some basal ganglia sitting in there, but that's the cortex. A tectum, we'll talk about the tectum of the midbrain. That's just the roof of the midbrain. I'll try to show you how that looks like kind of a roof. You have to imagine some of these things. Be creative with your imagination. Tegmentum means covering. We'll talk about the tegmentum of the midbrain too, which is a covering. So, all right. Questions on language? We're going to get into some anatomy now. This is like the big, you're going to hear all this stuff again 10 times during this course, okay? This is big picture. We're just trying to give you a big overview so we can at least start talking about getting more specific in different regions. So let's start with the cerebral hemispheres. These are the big guys that most people think of as the brain proper. What does it do? It processes lots of information: sensory, motor, memory, reasoning, language, intelligence, personality, emotion. Cerebral hemispheres do all that work, okay? 4 primary lobes, right? We know the lobes: frontal, parietal, temporal, occipital lobes. There's also sometimes referred to as a limbic or insular lobe. And we'll show you guys that tomorrow on the brains. We'll pull apart the lateral sylvian fissure, and you can see some more little wrinkles and gyri and sulci. That's the insular lobe, or the limbic lobe. It's even deep into there. So that's a 5th lobe. And actually, I'm going to show you guys, try to tomorrow, just go over to the virtual reality and just make sure you guys feel comfortable using the VR for the brain. And you'll see they actually have the limbic or insular lobe there. We'll point that out to you. We'll pull that apart. Lateral sylvian fissure, that's a big, deep fissure on the side of your brain here. It divides between your parietal, frontal, and your temporal lobes there. We'll show you that in a picture in a sec. Corpus callosum, these are just big features, major features of the cerebrum. The corpus callosum is this connecting structure between the 2 hemispheres, the corpus callosum. Communication. The surface is composed of the gray matter. That's a little couple millimeter thick layer of cortex. The white matter is the deep stuff, mainly axons, right? It's carrying all that information to and from the cortex for processing. And then we have some deep cell body structures, the basal ganglia, that are made up of the caudate, globus pallidus, putamen. Don't have to have all those memorized right now. Just we're going to do a whole lecture on the basal ganglia. They can be referred to as the basal nuclei or cerebral nuclei. You'll actually sometimes hear it that. I have a textbook that's 600 pages thick that all it is alternative names for all the different structures in central nervous system. Central nervous system alternative names. And so it lists all the names, and there's like 3 or 4 alternatives for almost every name. So we'll try to stick with what will be consistent with your book, but it's amazing. But if you're trying to read literature, you're trying to read other books, or if you're reading like a book out of the UK or England versus US, they'll use different terminology sometimes. So I'll point out occasionally when those are more frequently used, or commonly you'll see those. We're still in the cerebrum here. Cerebrum also, the diencephalon is considered kind of part of the cerebrum in general. And so that's primarily we're talking about the thalamus and the hypothalamus. Those are the 2 main structures of it. However, some people include also the epithalamus and the subthalamus. So really anything with an alamus at the end could be considered part of the diencephalon. However, most people talk about the diencephalon, and it means that these 2 things make up the major component of that. Okay, it's a major relay center in your brain. It's kind of like the traffic control center. If you want to talk to the brain or get up to the cortex, you got to stop by the thalamus first. Okay, that's one of the big roles of the thalamus. It's a relay structure. So all your sensory information goes through the thalamus, most of it, except for olfaction. It's the only sense that smell does not go through the thalamus. It goes directly to your cerebrum. So. But every other sensory information, or if you want to go talk to other structures in the brain, a lot of times you have to stop by the thalamus first. Okay, the hypothalamus is the master regulator of your autonomic nervous system. Okay, for maintaining homeostasis, eating, reproduction, and so that's in general what it does. The diencephalon is relay information, sensory and motor, and homeostasis and reproduction, eating, and growth. Okay, so autonomic, motor, and sensory. So it's important. The diencephalon does a lot. The epithalamus, we won't worry too much about. It's the pineal gland. It helps to regulate pituitary function and your adrenals. Okay, it also has to do sometimes with sleep. Melatonin is released there and helps regulate some sleep function. And then the subthalamus is an area just below, you have the hypothalamus, and just below the hypothalamus is a thing called the subthalamus, a little region just below the hypothalamus that actually is mainly connected to the basal ganglia in its role. It plays a role in movement, okay? But this is just showing you some of those structures, and I think it'd be easier to show in the VR tomorrow, so how these structures are layered in there. But the gray, that's your cerebral hemisphere area, the gray, and then the yellow structure here, everything in yellow is basal ganglia structures, okay? Then a little bit deep to that is this blue thing. You can kind of see it sticking out back here, but it kind of is in this region. A little deeper is the thalamus. So you got your cerebral hemispheres out here. You go a little deeper in, you've got your basal ganglia. A little deeper in, hiding just a little deeper in, is the thalamus. And between the thalamus is this little sliver of a space called the third ventricle, and that's right down the middle of your brain there. But so you have a thalamus and these basal ganglia and thalamus structures are all paired structures on each side of the brain. Okay, and I think the VR is really neat to be able to go in one time and actually just pull each thing out layer by layer. Do that once or twice, and then you'll feel good about how everything is sitting in there. It's hard to get an appreciation in pictures like this. I think this one that I wanted to show, this is the thalamus, hypothalamus, subthalamus sits kind of right in here, but don't worry too much about that at this point. Cerebral hemispheres. Again, we're still on cerebral hemispheres. Let's talk about lobes. Frontal lobe, everything in blue. Right there. Primarily, we're talking motor, personality. We're going to be real, just kind of big picture here. Okay, gross major functions. Start just knowing the major functions of these things before you start getting too deep. Temporal lobe, this stuff in orange right here. Memory and auditory processing and function. That's primarily what its roles are. Parietal lobe, this area in green. Somatosensation, so sensory of the body, okay, so the body, and association areas. Association means helps to integrate and associate information from multiple regions of the brain. Because you'll see, if we look at the occipital lobe back here, you'll notice the parietal lobe, its borders touch all the other lobes. So it's an integrative function too. It helps to take your somatosensory information from your body, plus your visual information, plus your cognition, and help bring it together to make you help understand the world, process that information, and have action and so forth that makes sense. Occipital, primarily vision. Okay, so that's all it shows there. And then if you take that lateral sylvian fissure, this big guy right here, and you take a little bit of these little tongs and you spread it apart, or tomorrow if you take your fingers and carefully spread that apart, you'll see more little gyri and sulci down here. That's the insular or limbic lobe. They don't know exactly the function it has, but it's probably more regulation of autonomic function, but also mainly related to emotional overlay, when you're nervous and you get butterflies and you get flushed face and so forth, that plays a role in some of that autonomic regulation, this insular limbic lobe that's down there. So let's get through the lateral portion of the cerebral hemispheres, and we'll take another break here. So now I'm going to go and start pointing out some of the major features. And tomorrow what we'll do is you're going to get this in real, you're going to get it laying there in front of you. I'm going to do the exact same thing I'm going to do now. I'm going to go through the structures, show it to you one time today. Tomorrow, we're going to go through those same structures while you're looking at it in real life, pointing to it as we do the same thing, okay? So we're going to go right down the list here of these structures, starting with cerebral longitudinal fissure. You can't see it on here. That's the thing right here. You don't have it unless you have two hemispheres. So when you have two hemispheres, that big cut down the middle, that's a cerebral longitudinal fissure, divides the two hemispheres. Let's start with the easy one. I'm going to change my color to yellow here. Come on, cooperate. Cooperate. So this guy right here, the lateral sylvian fissure, is that. Got it? Helps divide the lobes. Insular lobe underneath that. If you pull it apart, you'll see the insular lobe. The next major feature we usually try to find on the lateral surface is we got to identify our central sulcus. Right there. It's a little different on each brain. We'll show you how to find that tomorrow. There's a little characteristic. That central sulcus goes just over the top of the top ridge of the cerebrum, and it dives a little posterior. And that's how you kind of identify it. Does it go slightly over and dive a little posterior? If you're trying to guess between a couple of those that are going up there, that's the one. It's the central sulcus. Precentral, it's this one right here. That's the precentral sulcus. post-central sulcus is this one here, and you kind of have to sometimes jump a gyrus or two to connect the dots. Not on all brains, but this is the post-central gyrus on this brain. This is not a great, easy to see post-central gyrus. Some of your brains tomorrow have a much more clearly defined post-central sulcus. Sorry, not gyrus, sulcus. There's a thing called a preoccipital notch. It's right here. This is the preoccipital notch. This is the cerebellum. It's generally about, if you go about 2/3 of the way back on the cerebellum, there's a little spot here where it just comes up like that and it looks like a little notch there. That's called the preoccipital notch. We'll try to help you define it tomorrow. Precentral gyrus. So once we've got our central sulcus and our precentral sulcus, everything in between, that's our precentral gyrus. Precentral, right in front of the central sulcus. Precentral gyrus. And then if we have our central sulcus and our post-central sulcus, everything in between there is our post-central gyrus. So behind the central sulcus, post-central gyrus. The precentral gyrus, this is all motor, primary motor, precentral. All this post-central gyrus is primary sensory. Okay, that's where your sensory information goes from your body. Okay, and we'll talk about the homunculus, which is basically an overlay of your body's image on that area, and that's where the different parts of your body are represented from a motor and sensory standpoint, called the homunculus. Just point out here, there's a superior temporal gyrus right here. This is the superior temporal gyrus that I'm outlining. There's actually and these names are pretty easy sometimes to guess. There's a sulcus right here called the superior temporal sulcus. And there's another sulcus right here called the inferior temporal sulcus that divides our temporal gyrus into superior, middle, and inferior. But I only show you the superior because the important part here is this is where auditory information comes in right here. Initially is processed in the brain on that region of the superior temporal sulcus. I mean gyrus, sorry. That's where the primary auditory cortex is laid. And then from this, we can kind of do our lobes here. So if we find our central sulcus, our lateral sylvian fissure, our preoccipital notch, and then the other structure we have to know, we have to look at this side. There's a sulcus right here called the parieto-occipital sulcus. Where it comes over the top here on this ridge here, you connect the dots between this. So it comes over right here. You connect the dots between that and that. That defines our occipital lobe. If you take this lateral sylvian fissure to extend it out like that, now we've got parietal, temporal, and frontal lobes. It's kind of how we define the borders of those, by extending imaginary lines between those structures. Hanging in there so far? Any questions on that? All right, another break. Give you guys another 7 minutes or so. Go. Moving on to the medial surface, which is a little more intimidating, but come on, there we go. This is our mid-sagittal section. Let's just talk about the major things we're going to see here. First one is this guy. Nice big white matter structure, corpus callosum. Okay. Connects. It's a commissural structure. It connects one area, corresponding regions of the brain with the opposite side. So all the this is connecting frontal lobe regions of the right side of the brain with frontal lobe regions of the left side of the brain. You know, and this goes all the way from kind of towards the, it's mainly the parietal and frontal lobe regions that this is connecting from side to side. Okay, left and right brain, so there's some communication between the left and right brain. Interestingly enough, that structure, you can cut through that, right through the middle of it, and not really notice any deficits unless you do some very specific, unique setups to expose the deficits. We'll talk about that. We'll talk about what, because they do this in pediatrics and in kids sometimes who have intractable seizures. If you have a seizure on one side of the brain, the only way it can get to the other side is through the corpus callosum. And so if they want to limit the seizure activity, they'll just cut the whole corpus callosum so it can't spread to the other side of the brain. And you wouldn't notice any deficits unless you set up a few very contrived experimental procedures to show the deficit. And we'll show you that later on. It's kind of fun to see. The corpus callosum can be divided into some subregions here. The first is the rostrum. This little tip right here, this is the beak or the rostrum right here. Okay, even though it's not the most rostral, this is the most rostral limit, but it's this tip or the beak here is still called the rostrum of the corpus callosum. This section right here, this curved area, is the genu. Genu is bend, right? Kind of like genu recurvatum, that kind of stuff. It's a bending, so it's an angle there. So that's the genu. This more straight region here, that's the trunk of or the body sometimes, the trunk or body of the corpus callosum. And then the little ball here at the end, that's a splenium of the corpus callosum. You don't have to know that for tomorrow's lecture. You just need to go corpus callosum if I've got a line pointing to that. Now for the lab practical at the end, you will have to, I could just highlight and have, colored in a specific region saying identify that portion or be specific. If I say identify the structure, be specific, and I've got just a portion of it highlighted, then you got to be specific. Diencephalon. All right, so here we go. I'm going to outline the entire diencephalon. Is right there. That's thalamus and hypothalamus. But now I'm going to break them into the two structures. This region right here is the thalamus, and then this region here is the hypothalamus. There is a thing right here. So here's a thalamus. Depending on what cross-section you see, tomorrow's specimens you may or may not see this structure, is the intrathalamic adhesion. There's a little connecting stalk between the two thalami. There's a little sliver of space between the two thalamus called the third ventricle. You have to have the two halves of your brain together to create the third ventricle, because this little sliver of space, there's a little bridge between the two thalami that goes right through the middle of that third ventricle, this connecting stalk called the intrathalamic adhesion. Some people don't have an intrathalamic adhesion, and they don't have deficits, so they don't know exactly if it's really functional or really needed, or if it's an artifact of old evolutionary stuff of connection. But anyway, but you may see that bump on the thalamus, but you may not. That also depends on how good we were cross-sectioning it. If, as I cross-section through it, I might have been a little left or a little right and cut off that intrathalamic on one side of the cerebrum, but you'll see a big one on the other side, but not on the other side because I didn't get right to the middle of it. I will take out brains this module, a couple. There's a few that have some deficits. I think they have like strokes or malignancy or something like that. I will let you know. I'll time it on days or a time that's convenient for you. So if you want to come watch, you want to learn, you want to take a brain out yourself, I'll show you how to do it. It's a good time. But you don't have to. You don't have to see that. Anterior commissure. There's this little region right there is the anterior commissure. Remember, commissural fibers are white matter fibers that connect one region with another region. This is the anterior white commissure. It has fibers that go, here's the temporal lobe down here on this side. It has fibers that go to the temporal lobe, and then it has fibers that come over to the temporal lobe on the other side that's been cut off right now, which you can't see. So that anterior white commissure, there's some axons that go from one temporal lobe over to the other temporal lobe to help it communicate. I'm going to erase that again and just show you again. There it is. It's sometimes called the AWC, the anterior white commissure, the AWC. Kind of like versus just sometimes called just the anterior commissure, but sometimes it's called the anterior white commissure or the AWC. Parieto-occipital sulcus. This is probably the easiest thing to find on the medial surface. It's a really pretty deep, almost like a fissure, but it's still just a nice deep sulcus right here between the parietal lobe and the occipital lobe back here. Then we have the calcarean sulcus is the next thing, and that's this one right here. Calcarean sulcus is kind of right in this region. There it is, calcarean. That divides our occipital lobe on the medial surface into the lingual gyrus and the cuneus. This is lingual. Lingual means tongue-like, and I guess if you, that almost looks like a side shot of a tongue, I don't know. Lingual gyrus. Then this right here, this region is the cuneus. Okay? And I guess it's kind of cuneiform in nature, or look, or like the cuneiform bone in the foot. It's got a little wedge look shape to it. I don't know, but that's probably why they call it the cuneus. So that's those two regions. I pointed that out mainly because your primary vision. Why? This is really becoming an issue. I guess I should go Mac, right? I probably wouldn't have this problem if I had a Mac or an iPad or something. This calcarean sulcus, your primary vision, when vision goes from, it comes in, crosses, some of it crosses in the optic chiasm, but anyway, it goes to your lateral geniculate nucleus, which are little bumps on your thalamus. You gotta go talk to the thalamus first before you go talk to the brain. Then you go through the optic radiations and you go back to the occipital lobe, and where your primary vision first lands is right on the edges of the lingual and cuneus. Right in this region here is where your primary vision lands. Okay, it's right on those edges on that medial surface. Cingulate gyrus and sulcus. Let's draw that out. Cingulate gyrus is this whole thing. Cingulate C-shaped. See how it's kind of C-shaped? That's a cingulate sulcus. And then this whole area right here, this is all the cingulate gyrus. It's all C-shaped. Okay. These all play a role in limbic system function. Helps modulate some autonomic function, but also emotional responses and those kind of things. I don't think I have this on here, but this is just something to be aware of. There's a sulcus right here. See that sulcus that's right above the corpus callosum? Right below the cingulate gyrus, or between the cingulate gyrus and the corpus callosum. That's called the callosal sulcus. Callosal sulcus. The optic chiasm. So remember I said that some of your visual information crosses to go to the opposite side of your brain. And when we do their visual lecture, you'll see where that happens. But right now, there's a crossing point. So your optic nerves come in, they join up together. Some of that information crosses, some stays on the same side. But when we cut right down through the middle, your optic nerves are coming out here. We cut right down through the middle. That crossing point is called the optic chiasm. And that cut is right here. That white area right there, because this is white matter. These are axons. But that little round area there, that's the optic chiasm. This is actually the tag end of the optic nerve coming off here. It's going to go to your eyeball. So this area right here is the optic chiasm. There's a structure right down here that's almost like a little baby brother to the corpus callosum, but it's not really a commissural fiber. It's this little guy right here that's similar in shape. That's the fornix. We don't care about it as a physical therapist much. It's just another limbic system structure. That doesn't have a lot of discrete function to it, but it just plays a role in limbic system function overall. Again, that emotional, autonomic kind of stuff overlay. You can't see it here. There's actually this, see this little membrane right here? That's a membrane that's sitting right there. If I cut that off or flip that out of the way, you'd be looking into what's called the lateral ventricle. We have these spaces inside of our cerebrum that hold some cerebrospinal fluid. Each side we have two lateral ventricles with, so deep inside our brainstem, in there near that basal ganglia, the thalamic region, you usually have these ventricles, ventricular system. You have two lateral ventricles, that's your first and second ventricles, left and right lateral ventricles. They join and have little holes that go down into the third ventricle, which is the little space between your two thalami. And then there's a little aqueduct that goes down through the midbrain into the back part of behind your medulla, which is where the fourth ventricle lies, where your cerebellum. We'll show you that in a second. But this little flap of membranes called the septum pellucidum. It's like a septum in your nose. It's a septum. It's a division. But most of your brains won't have the septum pellucidum. Some of you may still have the septum pellucidum in there, depending on the cross section. I just point that out because most of you won't have that there. So you'll be looking right into the lateral ventricle there. You'll wonder what that space is. But there's a little hole right here. There's cerebrospinal fluid inside this lateral ventricle, and it comes out right here underneath the fornix through this little hole, and it's called the interventricular foramen. It's a foramen, a hole, foramen between the ventricles, interventricular, and then it gets in this whole region right here, which is basically your diencephalon on each side, but the space between them. That's the third ventricle. That's the space. If I said identify this space, not the structure. The structures are the thalamus and the hypothalamus. That space, that potential space, right now it's not a space because there's not another structure to give it that feature of a space. It's a potential space in there. That's the third ventricle. And then this is the little cerebral aqueduct right here. There's a little aqueduct that connects and then dumps into this space called the fourth ventricle back here. And then eventually your cerebrospinal fluid comes out from underneath your cerebellum to get into the subarachnoid space, which is a layer that covers, and then it gets in there to circulate all around the outside. That's how it gets from the inside to the outside of your brain then surrounds and circulates over the entire surface of your cerebrum, cerebellum, all the way down your spinal cord. Your brain and spinal cord and brainstem are all kind of like buoyant, neutrally buoyant, sitting in this cerebrospinal fluid. It gives a little bit of protection, a little insulation, a little nourishment. It's created there. Brainstem. This whole thing right here is your brainstem, right? If we go right here and kind of draw a line there, this is the midbrain. This region right here is the midbrain, which is up in the middle of the brain. This area here with this nice round protruding part, this is the pons. This round part sticking out is sometimes referred to as the basilar pons, that front protruding round part. And then down below here, till we get to the foramen magnum, this is the medulla. Those are your different components of the brainstem. And actually, I forgot to do this. Midbrain actually extends to this structures back here. There's this is all part of the midbrain. There's a couple little bumps off the back of your midbrain, and the cerebral aqueducts passing right through the middle of your midbrain. Those are also part of the midbrain, so sorry. This is actually referred to as the tectum of the midbrain, roof. It's kind of like it's sitting over the top of this part of your midbrain as a little bit of a roof or protective layer, but there's four structures there when you look at both sides of that, when you have the full midbrain there and looking at the back of it. But we'll show you that in a second here when we get to the brainstem. But so that's the tectum, but altogether that's the midbrain. Easy to see the cerebellum sitting back off here. I have to go look online, see if people are having the same trouble I'm having. So that's cerebellum. Just to highlight a few structures again, just because it gets confusing, especially in this region right here. There's a few confusing structures. Point them out again. Especially more specifically, this little region right in here gets a little bit confusing because we have the intrathalamic adhesion right there. We have the anterior white commissure right here. We have the optic chiasm right there. Then we have another structure I haven't mentioned because we don't really care about it that much, but it'll confuse you. It's this little bump right here called the mammillary body. You don't need to know that right now, but just know enough to not don't let it confuse you, you know. So intrathalamic adhesion right there, anterior white commissure, optic chiasm, mammillary body. Those little four structures can get a little bit confusing at times because they're so close to each other. They're just little. Inferior surface, which will not be on the quiz tomorrow. Any questions about that medial surface at the moment? Okay. Olfactory bulb and tract. Here's the olfactory bulb, and this is the tract. So remember, the tract is just the white matter. So on the olfactory bulb, you have some cell bodies. This is for smell, okay, the olfactory bulb and the tract. And remember, this goes directly into your limbic system. It actually goes into your frontal lobe and actually dives deep right into your limbic system. So smell is that's why it's considered a very primitive function. It goes right into your brain, right into your limbic system, and there's a lot of things associated with smell. It's a very primitive kind of sensory experience, and why it can bring back both bad and good memories and those kind of things, and have emotion, most of what you taste is smell. About 80 or 90% of what you consider taste is smell, and you know that when you have a bad cold and nothing tastes very good and you can't hardly taste a thing. And you're like, wow, I didn't realize smell played such a big role there. So that's the only peripheral nerve that goes directly into your brain without talking to the thalamus first, okay? Everything else has to go into your spinal cord or your brainstem and then go up and talk to the thalamus and then go to the spot in the brain. Olfactory sulcus. If you take that olfactory bulb and tract and just move it out of the way, like it's been kind of jagged up here and butchered a little bit here, there's a little sulcus that runs right there. That's the olfactory sulcus. It lays in the olfactory sulcus. Makes sense. Right here, this is called the straight or rectus gyrus. It's straight up and down, rectus, like your rectus abdominis is kind of straight up and down. It's called the straight or rectus gyrus. That is just on the inside on both your frontal lobes there. Here's the optic nerve right here, the tag ends of the optic nerve. This region here is the optic chiasm. However, there's a little guy in the way of our optic chiasm. There's a little structure, a little stalk right here, this little guy right there. That's the connecting stalk from your hypothalamus down to your pituitary gland. Your pituitary gland stays stuck in your skull. When you take a brain out, the pituitary gland is stuck in this little structure called the sella turcica. But anyway, so when you pull it out, that little stalk gets broken off. That's called the infundibulum. Don't have to know that, okay? But it's called the infundibulum. But it's in the way of our optic chiasm a little bit there. So what I'm really referring to is what's below that structure is the optic chiasm, is where some of your optic visual information crosses there at the optic chiasm. Once you've crossed the chiasm, got to figure this out somehow. Important won't do that. Chiasm, and then once you've crossed that, now these are the tracts back here, the optic tracts right here. Okay, just white matter fibers, axons, optic tracts. The parahippocampal gyrus. I'm going to show you the parahippocampal gyrus is this region right here. This is the parahippocampal. So this is parahippocampal gyrus. Parahippocampal, if you go deep in there, is the hippocampus. That's that big memory structure maybe some of you have heard about. Then the temporal lobes, the hippocampus is very important for memory, especially declarative memory, like memory you're trying to do now, memorize facts and things and those. This parahippocampal gyrus, if I took off the brainstem and the pons and moved it out of the way, it extends all the way like this. It kind of goes all the way back like this. It's called the parahippocampal gyrus from the inferior surface of the cerebrum. Hey, it worked. Let's see if I let it off. Okay, it worked. Who knows why? All right. Uncus. Uncus is this guy right here. That little the most medial protrusion of the parahippocampal gyrus is called the uncus. The only reason I mention the uncus is also coming out right here, and they're not there, is the oculomotor nerve. What happens is if you get a bad head injury, let's say you're got an athlete who got their bell rung, and let's say they do have a bleed or something, and there's some bleeding, and there's any kind of space-occupying lesion that's happening in the brain, it can push one side of the brain, and there's actually a membrane that you can't, the membrane's been removed, called the tentorium, okay? Covers over the cerebellum. It's kind of it's a dural membrane between the cerebellum and the bottom of your brain. There's this membrane, and it's called the tentorium cerebelli, the tent over the cerebellum. There's a little lip there, and that uncus gets pushed over the edge of the tentorium cerebelli, and it pushes on it protrudes out, hits the oculomotor nerve, and causes your pupil to blow out and dilate, because you lose parasympathetic control, and so your pupil blows out and dilates because you've lost the innervation of the oculomotor nerve into that particular eyeball. And that's why they're checking your pupils. Is this pupil dilated on one side? That's because your uncus is being pushed into your oculomotor nerve, because you must have significant enough space-occupying lesion that's pushing that uncus over the lip or edge of the tentorium cerebelli. And that's why if we go take a brain out and you watch me do that, you'll get that 3D, like, where's this tentorium cerebelli? How's it sitting in there? It's very, once you see it, you're like, oh, that's how that works. So anyway, that's just something. Don't worry about the anterior and posterior perforated substances. Don't have to worry about that. You really can't see midbrain much here. There's a little bit of midbrain right here and right there. And that very front part of the midbrain right there, those are actually, that very front part is called the cerebral peduncles. When you're looking at the outside of the midbrain from the front, we call it the cerebral peduncles right there. These connecting, that's what connects basically your midbrain up into your brain. But then this is the pons, and this is the front part of the pons. It's really called the basilar pons, that front rounded portion of the pons. And then here's our medulla right here. Now we're going to get into the brainstem a little bit more. Again, don't worry about just having all this stuff down. This is just getting you familiar with terminology, big overview. So, brainstem, 3 major parts: midbrain, pons, medulla. Okay? You see these bumps right here? Kind of hanging down there? Those are the mammillary bodies. And if now you might know why they're called the mammillary bodies if you use your imagination, or maybe so. And this is the front. See this? These right here. This is the cerebral peduncles right here. When you look at the front of the midbrain, and if I said identify these structure and be specific, that's the cerebral peduncles. Okay. This space right here is the interpeduncular fossa. It's just the space between the fossas, you know. I mean, between the peduncles, the cerebral peduncles. So interpeduncular fossa. That's your oculomotor nerve comes out of the interpeduncular fossa. So let's stop there. Medulla, some people use the formal long term, the medulla oblongata. Anybody seen the movie Gross Anatomy? I don't know if it's on Netflix or where you can get it, or Amazon Prime, but it would be a fun watch. It's like back from like the early 90s. It was like came out when I was in like PT school and we were going through anatomy. It's a pretty funny movie. It's about these medical school students going through medical school, and they're in dissection and gross anatomy, and there's a character who's got a strong Jamaican accent, and he calls it, the medulla oblongata, and he's just, anyway, so you got to see it. So do that one night when you just want to take a break. Look up Gross Anatomy, the movie, watch it. You'll get a kick out of it. It's where you're at right now in your life. It may not be funny at any other time in your life, but right now it might be funny to you. All right, a little more specifics here. Brainstem, ventral surface, front part, ventral or anterior surface. Okay, we're in the brainstem, so ventral and anterior match up here. Okay, I'll show you some cranial nerves briefly. We're not going to learn them right now. We'll just show you them on the next slide, so don't worry about it now. There's a little bit of a cranial nerve that comes from the back side. There's only one cranial nerve that comes off the back of your brainstem, and you can see a little bit of it right there. Don't worry about it. That's cranial nerve number 4. So we have the cerebral peduncles right here. Remember I said there's oftentimes more than one name for some of these? That's also called the crus cerebri or the basis pedunculi. The pedunculi at the base of the brain, the basis pedunculi, or the crus cerebri, okay? Or the cerebral, we'll call it cerebral peduncles. That's what it'll be on the exam, is cerebral peduncles. But these are fairly commonly used. So if you're just happen to be reading a journal article on the brainstem for fun, and they say crus cerebri, oh, that's just those cerebral peduncles. I knew that. We mentioned the interpeduncular fossa right there. Basilar pons, that's the round part. This is the basilar pons, and there's a little indentation or groove called the basilar groove. And you know what lies in the basilar groove, kind of creates that little bit of indentation, is the basilar artery. There's an artery that lays on the front of the brainstem, and we'll talk about that when we get to arterial supply. It lays in the basilar groove. Now down in the medulla, there's a couple things we'll just point out. These little bumps, there's a couple bumps on each side called the olives, and they look maybe olive-like shape, whatever. But they play a role in cerebellar function. When we get to the cerebellum, we'll talk about the olives. They supply some fibers up into the cerebellum to help regulate excitability of the cerebellum. Okay, that's the olives. Then we have the pyramids. So they're sort of triangular, pyramidal shape. Those are the pyramids right there in the front part of the brainstem. This is actually right underneath the pyramids is your motor tracts, motor fibers that run through the front part of your brainstem. So, erasing some of this. If I have some motor fibers coming from my cerebrum up here, they'll come down the front part of my brainstem. They'll come down in this crus cerebri right here. They'll come down through the basilar pons, the front part of the pons. They'll come down through the pyramids right here, the front part of the brainstem. And then eventually they'll cross over right about here. They'll cross over right in this region and then go down the opposite side to my spinal cord and then eventually to my anterior horn cells, to my muscles. So it's a contralateral pathway. Starts on one side, ends on the opposite side of the nervous system. But it goes down through the pyramids. And then the area where it crosses right here, this is called the decussation of the pyramids, or the pyramidal decussation. Decussation of the pyramids is this crossing point, because you have on the other side, you have these coming down this side, coming down this side. Yeah, this is not going to be good to do this when I get to the real complicated pathways. This is just the simple stuff, and it crosses over to the other side like that. And that crossing point is a decussation, okay? And the way you know, like if you've got an actual specimen of a brainstem, if you take your little probe or something and you stick it right here in this is called the ventromedian fissure right here of the medulla, and you just drag your probe down, eventually it'll hit a part where it kind of stops, and that's where those fibers start crossing. That's how you know you're at the decussation of the pyramids. All I want you to appreciate from this picture is just the things that are in yellow. Those are your cranial nerves coming off the brainstem, except for these are also cranial nerves. So you've got the olfactory and the optic as well that are not colored in yellow there. So, but those are your cranial nerves. They're all in pairs, right? There's ones on each side supplying different regions. The only one you can't see on here, well, you can see it actually. It's right here. This is the fourth cranial nerve that's coming from the back side of the brainstem. There's only one cranial nerve that comes off the back side. All the rest come off the front or the very side of the brainstem. There's only one that comes from the back side, and that's the trochlear nerve. It controls one of your eye muscles, okay? Superior oblique muscle. That's all it controls. It's like this little nerve that goes to that one muscle. Don't ask me why. Don't ask me why it comes off the back. But anyway. So now we're on the back, dorsal or posterior surface of the brainstem. So this is the back. This is right here. This structure back here is made up of there's 4 bumps back here. There's a superior colliculus on each side. So superior colliculus and then an inferior colliculus. Okay. There's actually some cell bodies underneath there, groups of cell bodies. These are relay centers. Some of your visual information and auditory information gets into these and gets relayed and processed for more reflexive function. So like when you hear a loud sound and it comes from this side of your body, when auditory information comes into your auditory nerve, goes directly into your brainstem, some of that information goes to the actually inferior colliculus, and then some of that information goes directly into your brainstem to tell your neck muscles to turn to the right. Before you even register that auditory information and to make sense of it at a conscious level, some of that information goes to your cerebral hemisphere, to that superior temporal lobe, to tell you what sound was that, you automatically, your head and neck will automatically reflexively turn to that loud, just to perceive threat, whatever, see what's going on over there. If there's a noise, that's mediated partially by that inferior collicular relay from information coming into the brainstem, goes to the inferior colliculus, then goes down into the brainstem to communicate with the upper cervical spine and neck muscles to tell them turn that way. Okay? We have that same function in vision too. The superior colliculi mediate that same thing. Even if you don't hear a sound and you see something in your periphery of your vision or something's coming at you as a threat from one side or the other, you're automatically going to look to that. You're going to turn your head, you're going to turn your eyes to that, and that is reflexively controlled partially through that connection with the superior colliculus. I generally think of my eyes a little higher than my ears, so that's how I remember the superior colliculus is visual relay for automatic kind of orientation looking towards things. And the inferior colliculus is my ears and auditory reflexive kind of things. As a group, people refer to this as the corpora quadrigemina. Corpora is body, quad, four bodies. There's 4 bodies. There's 2 superior colliculi, 2 inferior colliculi. It's the corpora quadrigemina. You don't necessarily have to know that, but just when you're reading and you see that, oh, that's just all the colliculi together as the corpora quadrigemina. Now, if we go back to this view right here, here is the superior colliculus right here and the inferior colliculus right here. We were looking at the back side of the midbrain. You see that? And then on the back side, looking at the surface of them, they're called the corpora quadrigemina as a group. When I'm looking at them in this view, I can either identify them as individually superior, inferior colliculus. This would be a right superior and inferior colliculus because this is the right side. Or if I identified this whole structure together, that's called the tectum of the midbrain. They form the tectum or the roof only in a sagittal cut like this. You call it the tectum because it looks like it's a roof over it. Here's that 4th cranial nerve, trochlear nerve, coming off the back side, right below the inferior colliculus. So that's a little tag end coming off the back. There's a fossa, and actually there's a little bit of a structure right here covering over the top part of this rhomboid fossa. This is the rhomboid fossa. This is actually called the superior medullary velum. You don't have to know that, but that's that little membrane there. Your cerebellum was sitting off the back here. The cerebellum has been cut off and taken off, and we're looking at the floor of the 4th ventricle. This rhomboid fossa or depression is forming what's called the floor of the 4th ventricle. When you put the cerebellum kind of forms the roof of the 4th ventricle. So if we go back to this picture, this is that floor. And if we're looking at it this direction and we're looking at the entire backside of a complete medulla, that's the rhomboid fossa we're looking at. Then you add, then that's the floor of the 4th ventricle. The cerebellum forms the roof, and then once it's there, when the cerebellum's sitting on the back, now you have a fourth ventricle. You have this space right here, which is your fourth ventricle. Does that make sense? But it looks like a rhomboid, okay? Rhomboid-shaped structure. A couple other structures here. When you cut the cerebellum off the back of the medulla, or pons, I should say, pons and medulla, it's right in that junction between the pons and medulla, you have these it has to be connected somehow. It's connected by peduncles. Peduncles are connecting things, right? Like cerebral peduncles. Well, these are cerebellar peduncles. So cerebellar peduncles connect the cerebellum to the back of the brain stem, okay? And you actually have 3 of them. But in order to see the 3 or really appreciate the 3, you have to get the angled cuts just right. And so let me show you. There's a superior cerebellar peduncle, a big middle cerebellar peduncle, and an inferior cerebellar peduncle, okay? And this is inferior because the very lower tip of it does actually go the most inferior. This is a superior cerebellar peduncle and a middle cerebellar peduncle. Those contain axons that are going to and from the cerebellum to the brainstem, okay? We'll get into what's going through those peduncles when we get to the cerebellum, okay? Information going from the cerebellum up to the brain tends to go through that superior cerebellar peduncle to go up to the brain. Information coming from your spinal cord that's going into your cerebellum is coming in through that inferior cerebellar peduncle. And then information that's crossing, that's going to go, is through the middle cerebellar peduncle, which we'll talk about later. But just know that there's 3 connecting peduncles, superior, inferior, and middle. All right, now moving down into the medulla. Medulla starts right here, right below that inferior cerebellar peduncle. You have a couple bumps on the back here, and you have to kind of almost imagine them. It's easier when you're seeing them in a real, like, 3-dimensional specimen. You can see the bumps. But there's a bump right there, and a little bump right there. Bump right there, and another little bump right there. The ones that are more lateral out here, those are the cuneate tubercles. The ones that are more medial right here, that's the gracile tubercle. Okay? Gracile, cuneate tubercle. There's little groups of cell bodies in there. Okay? And there's actually, you can take this back part and divide it into, you can see some little striations that divide this into 4 little columns. There's the fasciculus gracilis, lateral on both sides, and I divided that into too many sections. Hang on. Oh boy, this is going to be tough. And I don't know if it's the Snagit software interfering with the PowerPoint software or what's going on. But so let's say here's a central sulcus here, fissure. We'll divide it into that and divide it into that. So out here is the fasciculus gracilis. Fasciculus cuneatus. Fasciculus cuneatus, most lateral. And then this is the fasciculus gracilis back here. Okay. So those are axons that are coming up. If you look deep in there, if you cut through those and look deep in there underneath those fasciculi, gracilis and cuneatus, those are axons that are bringing in proprioceptive information from your body. The fasciculus cuneatus brings it in from your upper body. The fasciculus gracilis brings it in from your lower body. And I think gracilis muscle is in my leg, so that's how I know its lower body is gracilis. It goes up, and those axons are carrying that information from your body, and then it makes a connection with some cell bodies in either the cuneate, nucleus cuneatus and nucleus gracilis, which are just below those tubercles. The tubercles are the bumps on the outside. It's a surface feature. But just deep to those is the nucleus gracilis and nucleus cuneatus that are underneath the tubercles. Does that make sense? So in a surface structure like this, you're just going to refer to it as the tubercle or the fasciculus. But in a cross section, That's when we'd say it's a nucleus gracilis, the nucleus cuneatus, versus it's not the tubercle anymore. It's in, but again, don't have to know all this right now. I'm just starting to tell it to you the first time, so when the fourth time you hear it, it'll finally click, okay? So it's okay if this is overwhelming you a little bit right now. Tomorrow's quiz, you just got to know that lateral surface of the cerebrum and medial surface of the cerebrum, those 10 or 12 things on each of those, cerebellum, let's just kind of skip through this pretty quickly, just if my thing will let me. Cerebellum, that's the whole cerebellum together. It's got an anterior lobe, which is this region right here, and then this big posterior lobe right here. So anterior lobe is about 1/3 of your cerebellum. The posterior lobe is more like 2/3 of your cerebellum. This is looking at the anterior, surface, the anterior ventral surface of the cerebellum when it's been cut off of the back of your brainstem. And you remember that we had those peduncles, those cerebellar peduncles, superior, middle, and inferior? Well, here they are. This is the superior cerebellar peduncle. This is the inferior, and this is the big middle cerebellar peduncle. Superior, inferior, and middle cerebellar peduncles, where they've been cut off the back of the medulla and pons area. This is looking at the superior or dorsal surface of the cerebellum, because the cerebellum is lined up with the cerebrum in this. So superior or dorsal are synonymous here. So we're looking at the superior dorsal surface of the cerebellum here. We can divide the cerebellum into some like vertical divisions, either in a frontal view or a dorsal view. We can divide that into vertical divisions, which the very middle, this is called the vermis. Vermis means worm-like. It's kind of like a little worm-like, and it looks a little more worm-like right through here. The vermis is this more most medial port. Then we have what's called the paravermal region, which is right out here. Paravermal regions are out here. And then these big lateral hemispheres are that's the lateral hemispheres. Got it? Getting there. Spinal cord. You guys know this stuff pretty well, I think. So we start cerebrum, brainstem, now we're in spinal cord, or we did cerebellum, spinal cord. Lies within the vertebral columns, within the vertebral column, the spinal canal. Extends to level L1, L2. So your spinal cord doesn't go all the way down. It stops between L1 and L2. And that has some clinical significance for us, which we'll talk about when we get to the spinal cord. The end of the spinal cord, where it stops at L1, L2, there's a bunch of nerves coming off that need to still go down and go all the way down to the lower levels of your spinal cord, to the sacrum and lower lumbar segments. All those little nerves coming off are called the cauda equina, horse's tail. It looks like a tail, kind of like a horse's tail. You have these dorsal and ventral roots that come off your spinal cord that join together to form the spinal nerves. The dorsal roots are afferent. That's where sensor information comes in the back part of the spinal cord. Afferent neurons are bringing in, it's an afferent structure, the dorsal roots. The ventral roots are motor. That's where the motor function goes out. But they all come together at the spinal nerve. So once you get to the spinal nerve, you have motor and sensory together. Plus, if you're in the thoracolumbar region, you also have some autonomic, sympathetic fibers in there as well. 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, 1 coccygeal, paired nerves. These are all pairs of nerves. That's why you get the 31 spinal nerves. All right. So that's just showing you a sagittal section and a frontal view of the spinal cord. Here's the tip of the spinal cord. Let me, if I need to go a different color here, maybe a little darker colors. Tip of the spinal cord is right down here. And then here's all the spinal nerves that still have to go all the way down and exit out their appropriate levels that are coming off the tip of the spinal cord that forms this cauda equina, the horse's tail. This little very tip part right here, the spinal cord, that's the conus medullaris. This little cone tip, conus medullaris. This is an actual specimen here. You've done like laminectomies all the way down the spinal cord. But this is right here, the end of the spinal cord. This is between L1 and L2. This is the conus medullaris right there. Here's your cauda equina. And there's this little tiny, can you see that little tiny structure? It's a little lighter in color right in the middle there. You guys can see that there, that little structure that's a little bit lighter in color right down the middle. Then it goes right down there. That's the filum terminale. It's this little projection of pia. Pia is a little layer of, it's a layer on top of your nervous system. You have pia over the entire surface of your brainstem. It's one of the three meningeal layers. You have pia, arachnoid, and dura that form the pad, PAD, pia, arachnoid, dura. But pia is this microscopic layer that you really can't see until it actually projects off the tip of the spinal cord and actually forms multiple cell structure that projects down and anchors your spinal cord to your sacrum. And that's the filum terminale that does that. And that's where, like, you know, when like you've heard of craniosacral therapy and all this stuff, where they like say they can feel the movement of your sacrum moving in conjunction with your cerebral spinal flow, and they're gonna hold their hand there and help reset it, and then by doing that helps to keep the filum terminale from tractioning on your dural structures and whatever. I mean, there's some, you know, there's maybe something to that, and there is an actual physical connection between your sacrum and your spinal cord through this filum terminale. And there's a syndrome called tethered cord syndrome, where some children, like their vertebrae grow faster than their spinal cord lengthens, and it yanks on the filum terminale and tethers the cord and causes neurologic dysfunction when their vertebral column growth outpaces their elongation of their spinal cord, and that filum terminale tugs on it a little bit. So, internal features of the spinal cord. So we're looking at a horizontal section through the spinal cord here. Just know that the stuff in the middle is the gray matter. That's where the cell body is. So what's unique about the spinal cord is all the gray matter's in the center. Where in the cerebrum, most of your gray matter's on the outside, in the cortex. Except you do have those basal ganglia structures. You do have a few nuclei deep inside the brain, but in the spinal cord, most of the gray matter's in the middle, forms that H-like structure. You have the dorsal horns and ventral horns. So this area here, these are the ventral horns, and then these are the dorsal horns. In your thoracal lumbar region, where you have some sympathetic neurons, you will also see a little bit of a lateral horn. Okay, there's some cell bodies that are that form your sympathetic nerves that are in that lateral horn there. Okay. Your anterior horn is where your cell bodies that are going toward your motor cells for your muscles. Sensory information comes in initially to the back part of the spinal cord, interacts with cell bodies that are in the dorsal horn. Okay. Looking at the spinal cord just in a very gross structural way here, remember I said this whole region right here, this is called the posterior funiculus. This is the posterior, that's back part. This is the anterior funiculus, and this is the lateral funiculus, so these big white areas there. But within, so this is the posterior funiculus. The posterior funiculus is divided into some posterior columns. We're not going to go into that. You can subdivide those bigger funiculi into smaller columns of these are all axons, white matter, running up and down the spinal cord, taking information to the central nervous system or bringing it down to the spinal cord. Okay. This is it laying within the vertebral canal. This is showing you, the dorsal root right here and the ventral root. So the motor activity and motor cells, and also the autonomic. If there's sympathetic neurons, they go out the front part right here. And then eventually these join up together to form the spinal nerve here. Information that comes in the backside is going in to supply sensor information through that dorsal root and then into the dorsal horn. But right here, once you get to this point, everything's mixed. And if here's some sympathetic neurons, they'll come in here. So once you get to the spinal nerve, once you exit out the foramina there, the foramina, and joined to form your spinal nerve, everything's mixed. Okay, and pretty quickly your nerves start dividing into like a dorsal rami that goes to the back part of your body, that goes to supply the muscles and skin of the back part of your body. This actually goes out to form the ventral rami that goes out to the front part of your body to supply the skin, muscle, all that of the front part of your body. But you don't have to know yet. And we are going to stop right there. So tomorrow we got early morning, fun times on Tuesday mornings, but at least then you're done early. You got at least a day to kind of do some other stuff, sleep. Tomorrow we will finish up